is that the viral Sark gene, a prominent difference between viral Sark and cellular Sark is that the viral Sark gene is missing this carboxy terminal domain entirely. What effect would that have? And why? Yeah. It wouldn't be phosphorylated, yes. So it's missing the tyrosine and it won't be phosphorylated. And so what's the consequence of that? That's good. Yeah? It wouldn't be inactive, meaning it would be active. Excellent. Okay. So if it's missing this phosphotyrosine domain, the SH2 cannot fold properly by binding to what's missing, right? If it can't bind the phosphotyrosine because it's missing, you can't have this folding. And as I mentioned, the unfolded protein becomes active, okay? Because the folded structure is inhibitory to the catalytic activity, and the unfolded structure frees the catalytic domain to be fully active. Okay. So the predominant mechanism or mutation which has caused viral Sark to become hyperactive or fully constitutively active, continuously active as a protein kinase is the loss of the ability for this folding to occur. Okay? So what does that suggest about how C-Sark might normally be activated? Yeah. Sorry? Okay, excellent. So one possibility is dephosphorylation. So we'd like to know what is the mechanism by which this tyrosine is kept phosphorylated. And it turns out there's another protein kinase called CSK or C-Sark kinase that phosphorylates constitutively in normal cells, constitutively phosphorylates this tyrosine. And if we remove CSK from cells, that tyrosine becomes unphosphorylated and Sark becomes activated. So one way of activating this kinase is by dephosphorylating this or preventing its phosphorylation. Okay? What's another way that we might activate this protein? Yeah. Sorry? Okay, well that's what, cleaving the C-terminus is what has happened. So clearly that's a way. What's another way? Yes? Okay, changing the tyrosine, so a mutation that might change the tyrosine to another amino acid would do it. That's good. What else? Stop thinking about the C-terminus. What else? Yeah. Sorry?
Extreme conditions like extreme heat will unfold the protein.
Okay, he's saying extreme conditions that would unfold the protein. We don't need extreme conditions. We can unfold the protein in very specific ways. That's good. How? Yeah. Sorry? Okay, that's an extreme condition, would be removal of the SH2 domain. Good. Yeah? Okay, good. So mutations, additional mutations that might interfere with this folding process, or really anything that would interfere with the normal folding. If we disrupt the SH2 interaction with the phosphotyrosine, if we disrupt SH3 from interacting with the kinase domain, if we disrupt the folding, we cause an unfolding of the protein, and anything that would cause an unfolding of the protein would activate it. Okay, so C-Sark can be activated by a variety of possible measures that would cause a disruption of the normal folding. We could disrupt the SH2 domain interaction, disrupt the SH3 domain interaction, mess around with that phosphotyrosine. All of those will cause an activation. Okay? And what we've seen is that the activation, the major activation mechanism for VSARC was the removal, the truncation of that whole terminal region containing the phosphotyrosine. So now we understand we've uncovered a whole new class of protein kinase that plays a critical role in cell growth control. We can mutate this protein and cause it to become an activated protein kinase, which now acts in a dominant way to cause uncontrolled growth. Now, what do I mean by a dominant way? Well, if I have Rous sarcoma virus, which has the VSARC gene, or I just isolate the VSARC gene and introduce it into cells, those cells have a normal cellular SARC gene. But by introducing this activated form of a mutated gene into cells, it acts in a dominant way over normal growth control to cause uncontrolled growth. So it's a dominantly acting mutant oncogene that acts in a dominant manner on the growth control process and is dominant over the proto-oncogene CSARC. Okay? I know that mutations, a variety of mutations that I could introduce into CSARC would cause it to become oncogenic if I could cause uncontrolled growth through unfolding. One of those type of mutations is what VSARC has done, but any of the disruptions of the folding would also achieve similar effects. Now that leads to a very important and interesting hypothesis, and that hypothesis is, or the question is, that cancer may be caused, in general now, cancer may be caused by a mutation of a cellular gene that causes it to become dominant over the normal growth control process, such that you have abnormal growth control. Does everyone get that? So the question is, by example of what has happened with Rous sarcoma virus and cellular SARC and VSARC, one may make the hypothesis that cancerous growth could be caused by mutation of a normal cellular gene, causing it to become uncontrolled and dominant over normal cellular growth control process, such that you get uncontrolled growth. So growth is normally governed by a growth control pathway, and if I take a component of that growth control pathway and I disrupt it and make it, in the case of SARC, hyperactive as a kinase, I will now cause uncontrolled growth. That was the question. So we're going now from the example in a virus that causes tumors in chickens, Rous sarcoma virus, and identifying the genetic basis of that and the biochemical basis of that, and seeing whether that reflects a general understanding of growth control processes in human cells underlying cancer. Yes?
You said that it controls the growth. So if it controls the growth, then their cells will be replicating more through mass action in comparison to other cells. So it would just be more replication of DNA. So once it's replicating the DNA, that protein will also be replicated. So then if you just keep on doing sequential steps, then eventually that won't be the most dominant piece of culture.
So we don't need to think about populations of cells in order to understand the dominance. It's really at a molecular basis in a single cell that we're talking about dominance. Okay, yeah.
Is it like more stable? Is that mutant more stable than the regular configuration?
Is the mutant more stable? The mutant is active, and the normal cellular gene is not active. And by having this constitutive activity,